Hello everyone, today we're going to be talking about prokaryotic cells. So in that we're going to be talking about prokaryotes. So what exactly are these types of cells? So when you look at prokaryotic cells, what makes them stand out, or what makes them unique, is that they don't possess a true nucleus. So our first point is that they don't possess a true nucleus. This is to say that the genetic material is not enclosed in a nuclear membrane. For instance, this is a nuclear membrane, all right? And this is the genetic material. So what happens is that in prokaryotic cells, this genetic material is not found enclosed by a nuclear membrane. So this is what they mean by it does not contain a true nucleus. However, when you look at the genetic material, for instance, if this is our genetic material in prokaryotic cells, you're going to find that it's going to be found freely floating in cytoplasm. So you find this in cytoplasm. Okay, so we're going to see more of these as we look at certain drawings and pictures. So the best prokaryotic example is of bacteria, which I drew here. So this is an example of a bacteria such as E. coli. So you look at the parts that it has. This is the nucleus that we said is found to be freely floating in the cytoplasm. The other parts that we have flagellum, we have the cell wall. This entire thing here is the cell wall. Okay, then this is a capsule. This then this that you see here is Pili. Then you have the mRNA, which is also genetic material, as well as the polyribosomes. Polyribosomes because it's a long chain of ribosomes attached, hence that's where the name comes from, the polyribosomes. Okay, so next up we're going to talk about the cell membrane. Okay, so what exactly is the cell membrane? So the bacterial cell is surrounded by a partially permeable cell membrane like we had seen. Let me just go back to the image. I think it was highlighted. So this that you see right here, this inner coating right here, this is the cell membrane. Okay, now something unique about it is that it's actually semi-permeable. Okay, there we go. So our keyword here is that it's semi-permeable. So what do they mean when they say it's semi-permeable? So they simply mean that this does not allow everything to pass through it. So what you find out is that only a few substances are able to penetrate through. So are able to pass through. That's why it's semi-permeable. Another thing that you'll notice is that it's actually a site of respiratory enzymes. Okay, so it's a site of respiratory enzymes. So in some bacteria, the cell membrane can also be used for photosynthesis. Okay, which in layman's language is the process by which plants can make their own food. So just use simple language. Then when you look at the cell membrane, it's mainly composed of a lipid bilayer. So if you have watched the previous video on lipids, we talked about a lipid bilayer. For instance, this is it's called a bilayer because it's got two layers. Okay, now why does it have such a shape? It's because one end is hydrophilic, the other end is hydrophobic. So the end that is hydrophilic is the one that sticks out. The one which is hydrophobic is this part, is the one that sticks inside. So this is hydrophobic, meaning water hating. So this is what they mean by it contains a lipid bilayer. Now within this lipid bilayer you find several things like carbohydrates, other things like phospholipids, okay, and other things which I'm just going to draw here. You're going to find within it is what we call an integral protein. Okay, so these are integral proteins. Some of them act as transport proteins, able to they help the bacteria to transport certain things from outside to the inside of the cell. Then other things that you can also find, you can find cholesterol just within there. You're able to find cholesterol. Okay, so the first thing we talked about is that you can find respiratory enzymes, meaning respiration is helped just there at the surface of the cell membrane. The other things that you can what you observe is that cell photosynthesis actually occurs on the layers of the cell membrane. And another thing is that the cell membrane is called a lipid bilayer. Bilayer because it has two layers. This is the second layer, this is the first layer. And in between the layers, we've said you can find integral proteins, you can find cholesterol, you can find carbohydrates, you can also find phospholipids that are part of the bilayer. Another thing that we are going to talk about is the pili. Pili is plural, singular is pillars. So what exactly does the pillars do? So let me go back to the image that's here. So you look at these hair-like strands that are found on the bacteria. So what happens is that the reason why bacteria has what we call pillars. So let me draw this is one bacteria. So this is pili that projects onto another bacteria, and this is the way bacteria is able to exchange genetic information. Okay, so bacteria exchanges genetic information, thereby undergoing reproduction. So like we said, it's a hair-like protein structure. So these are actually protein in nature. These that you're able to see right here, these are protein in nature, and they stick on the surface of the bacteria, enabling it to stick to another bacteria for the purpose of exchange, like we just mentioned here. The next thing we're going to talk about is flagellum. I'll keep on going back to the image. Like you saw, this is flagellum right here. So what exactly is the reason as to why bacteria has flagellum? So I'll draw it once again here. So flagellum, I just drew one, but it can actually have several flagellum. So it's made in such a way that it's able to propel this bacteria and enable it to undergo locomotion. So it's able to move from one place to another. So this flagellum is made of a complex structure or a protein that we call flagellin. Okay, so it's made of a structure that we call flagellin. So it looks like a whip. You know what a whip looks like? A typical whip. So this is what flagellum looks like. It's a whip-like complex structure made of a protein that's called flagellin. So it helps bacteria move. So bacteria is able to move around by the use of the motor protein that spins. So this flagellum spins as the bacteria is moving from one place to another. It spins around and enabling it to move. So the base of the flagellum rotates in a ring-shaped. You know what a ring looks like? It looks like this. So when it's rotating, it rotates in a ring kind of motion, so that the flagellum performs rotational motion and thereby enabling this bacteria to move forward. So this is how bacteria is able to move, by virtue of it having a flagellum. The next thing we're going to talk about is the cytoplasm. So the cytoplasm is a fluid that's contained within the bacterial cell wall. So like you can see, this is the cytoplasm. Okay, so the cytoplasm is just simply a medium. Okay, it's the fluid that's contained. So the first thing we'll talk about is that it's a fluid that is contained and it's enclosed by the cell membrane. Okay, so for instance, this is your bacteria. So the fluid that's found in here is what we are calling the cytoplasm. Okay, and it's bounded by this cell membrane right here. Okay, so in this cytoplasm, it contains several things like ribosomes, where protein synthesis occurs. You have DNA, you have enzymes, you have lipid droplets, like you saw. So this liquid itself is cytoplasm. Okay, and you can see that there are several things that are found swimming within the cytoplasm. So this is to tell you that there are so many cell processes that occur within the cytoplasm of bacteria. Okay, one of the integral things we actually talked about is that prokaryotic cells don't have a true nucleus. So how then do they have genetic material? We said it's found freely floating in the cytoplasm. So a nucleoid is the region in the cytoplasm that contains the secular DNA. So there is secular DNA. Okay, so that region that you're able to find this secular DNA, okay, is what we are referring to as the nucleoid. Okay, and remember that this DNA is double-stranded. So this nucleoid is able to store genetic information. Okay, so it stores DNA that has genetic information that's encoded. So this hereditary information is one that controls the cell, and it can be passed on to daughter cells. Okay, so the genetic information can actually be passed on to daughter cells. So bacterial chromosomes are not, the bacterial chromosome is not enclosed in any membrane like you can see here. We said it's found freely floating within the bacteria, but it just exists as it is. So what we're simply trying to say is that this nucleoid is just a place where you're able to find this secular double-stranded DNA that contains genetic information of that bacteria. So the next thing I'm going to talk about is plasmids. Okay, and once again I'm going to go back to that image. Okay, so a plasmid is like you can see from the diagram, is this one right here. Let's extend this arrow. There we go. So this is our plasmid. So let's just go back to our drawing board. So what exactly are plasmids? So these are small pieces of DNA. So like they look like small fragments of DNA which are separate from the chromosomal DNA. You remember that secular DNA that we had seen found in the nucleoid? For plasmids, the small secular DNA that are found away from the chromosomal DNA. So they usually replicate at the same time. So when this DNA is undergoing replication, which is it forming a copy of itself, these plasmids, they undergo replication at the same time as the secular DNA, in as much as they are separate from each other. They undergo replication at the same time. So, but they're not essential for the survival of the bacterial cell. Okay, see that the small fragments. So the most important thing that helps the survival of this cell is the secular DNA. Okay, unlike these plasmids that are just found floating and are separate from the chromosomal DNA. Okay, the next thing we'll talk about is ribosomes. Okay, so ribosomes are also found in what we are going to be learning next, which are eukaryotic cells. But let's just elaborate more. So ribosomes, these are a site for protein synthesis. Okay, so simply, let me give an example. This is a ribosome. So the ribosomes are found in what we call Sverdrup units. So these are just like subunits. They don't really add up additionally, but they are just called subunits. For instance, for the bacteria, it has what we call the 70S ribosome. So this 70S is for this whole entire ribosome, and like I said, they are not addition, they don't add up. For instance, the large subunit is what we call the 50S. Okay, the S stands for the Sverdrup unit subunits. Then the small subunit consists of 30S. Like you can see, when you add 30 plus 50, it doesn't give you 70. It gives you 80, which is what we mean by they are not additional. So when you look at these subunits, they come together in a process of what we call protein synthesis. So they help in protein synthesis by bringing together the mRNA, which is messenger RNA. Sorry, let's just erase that. So the subunit is simply like a factory, okay? So this is where everything takes place, the protein synthesis. So the messenger RNA comes in this place, and also the tRNA, which is the transfer RNA, which is the transfer RNA. All these help in what we call protein synthesis, okay, which I won't talk about more in details because, you know, tRNA has a shape of a cross, okay? So this tRNA carries genetic material and brings it to the this ribosome, okay? So it carries them and it helps to encode the genetic information that's found on the messenger RNA, okay? So for more details, you may go ahead and watch the video that was made concerning protein synthesis as well as DNA replication. So this is what exactly it's about. The next thing that we're going to talk about is glycogen granules. Okay, so I missed a word. Let's just add that. So we have glycogen granules and lipid droplets. I'm sure you had seen some of them in the previous photo. So these are the small granules, okay? These very tiny ones right here. These are the granules, the small ones right here. And you also have the glycogen granules and the lipid droplets, which we didn't quite mention, but are also found within the bacteria. So when you look at these glycogen granules, what exactly are they? So these they appear as inclusions. So the key word here is inclusions, okay? So they appear as inclusions in bacteria, in the bacteria cell, and they are used to store energy, okay? They store energy, which actually becomes useful when the bacteria is experiencing starvation, okay? So they act like food storages, the glycogen granules and lipid droplets, just like a human being. We have glycogen, you also have fats, you have proteins. So what the body does is that when you've run out of glucose, it uses up the glycogen stores. When the glycogen stores are done, it uses up the fats. When the fats are done, it uses up protein. And that's how come people are seen to actually lose weight because the body keeps on using up their body mass in order to give them energy. So the same thing is being used in a bacteria. So how does it store energy? We've said that it has glycogen granules. The next and the last thing that we're going to talk about, which is very important, just like the others, is the cell wall. Okay, let me go back to our image right here. So the cell wall, which is here, this arrow. So the cell wall, like you can see, surrounds the entire cell. Okay, so the cell wall gives rigidity. So these are keywords that you need to take note of. So it gives rigidity to the cell. Okay, instead of bacteria just being easily squashed rather or destroyed, it gives it the ability to have rigidity and maintain its shape. Okay, so it prevents, for instance, when water is coming into the cell, it prevents the cell from actually bursting. Okay, because as there is water, there is also an inward force that is pressed on the cell wall, thereby when the two pressures equalize, then there will be no net pressure, which is going to enable this to burst. Okay, so as water is coming in as a result of osmosis, this bacteria won't be able to burst. So water and various ions, okay, water and various ions and small molecules can actually pass freely. For the cell wall, it's not like the cell membrane where it's semi-permeable. This one can allow all types of substances to go through it. Okay, but larger molecules such as proteins, so let's jot them down. Larger molecules like proteins, nucleic acids. So you have nucleic acids. These aren't able to pass through the cell wall by virtue of them having big molecules. So the cell wall has a substance that we call neurine. Okay, so it has a substance that we call neurine, consisting of a polysaccharide chain. So neurine consists of a polysaccharide. So for instance, these are monosaccharides. It's a long chain of monosaccharides, right? This is what happens. And another chain, these are polysaccharides. So this substance is what we call neurine. Okay, so these polysaccharides, they are crossed, linked with short peptides. So for instance, if this is a short peptide that's holding these two polysaccharides together, okay, so these are peptides. Okay, now these peptides, they form cross-linked that are able to hold the these individual polysaccharides together. And that's why when you go further to learn about gram-positive bacteria, gram-negative bacteria, they simply mean for gram-positive bacteria, it's bacteria, for instance, like lactobacillus. They mean it's surrounded by a cell wall such that when you use a gram stain, it stains the bacteria and you're able to pick that, oh, okay, there is presence of a bacteria here. But for those that are gram-negative, they're quite difficult because you can't stain them because there's nothing to stain about them. Because what we're targeting is a cell wall. But if the cell wall is not present, it means you can't stain that bacteria. Hence, we call them the gram-negative. Okay, gram-stain negative because they can't be stained by ink. Okay, so the whole moral of our lesson today is that we actually, as you go further in your study, you realize that there's a reason why we actually learn about prokaryotic cells, bacteria, because certain drugs will be targeted against destroying the cell wall of the bacteria. Others will be targeted to destroy the cell membrane or the nucleus of that bacteria. So that's why it's important to know what that bacteria consists of, because if you create a drug that destroys the cell wall, meanwhile that bacteria is gram negative, it means that drug won't work. Okay, so that's why we had to go through this lesson. Thank you so much. I hope you enjoyed the class.